data. Okay. So there's like different technologies and modalities for spatial data, each having their own pros and cons. Like for example, Visium is a platform where you have spots in a grid, and you're able to have a bunch of genes for that. But the issue with that is it doesn't give single cell resolution. Um, Xenium is another very popular platform that has single cell resolution, but you have a very limited gene panel. You have about like 280 to 5,000 genes for that. So that makes spatial analysis a lot more difficult than like just doing simple like pipelines and stuff like that. So current downstream methods identify niches as groups of cells that are spatially localized that may perform a function via cell-cell signaling. And an example of this is a tool called SC that takes spatial data as input, and it extracts three different features: the molecular profile of the cells, the molecular profiles of the cell neighborhoods, and the overall cell composition of each of those neighborhoods, and kind of uses that to form these groupings of cells. And methods like these are cool in terms of they can find these localized, spatially localized cells, but they don't really tell us too much about the functions that they might perform together. So yeah, the current issue with the tools are they take similar approaches to creating niches based on spatial data or some latent space, but it doesn't ask the question, how do these niches communicate with each other? Like, for example, if we have a tumor sample, how does the stroma or the cell surrounding the tumor actually affect the tumor? And vice versa, how does the tumor send stuff out to the stroma? And how can we quantify the function that a niche, for example, performs? And so I created this method, but before we get into that, I want to kind of give background into the tissue that I will be kind of using as a biological example for this. From Kai's lab, we obtained human breast tissue samples of healthy patients, pretty much so we can benchmark, is this supposed to be happening? Because there's a lot of studies with breast tissue and the type of signaling and pathways that go along with it, and the types of groups of cells that uh, are supposed to be localized in space and through expression. And so in breast tissue, the three main structures are the stroma, the lobular units, and the ducts. So the stroma is kind of the tissue surrounding the structures, like the ducts and the lobular units in the breast. And the lobular units are these alveolar-like structures, like similar to the lung, that contain epithelial cells and diverse that perform functions within the breast. And then the ducts are like the branch-like structure that connects the lobular units to the nipple, ultimately for lactation purposes. Um, and as a basis for my method, which I'll get into, we used publicly available methods from Qing's group, specifically Comet and Spaceflow. So Comet, what it does is it uses optimal transport as a way to predict how cells interact within a tissue space, or if you're looking at Visium data, how do two spots in the space interact with each other. And it uses a gene expression, spatial distance, and downstream analysis to look if it's causative or like coincidental. And so for a little background on optimal transport, it's like this mathematical framework used to align two probability distribution functions. So like ligand A can is expressed at this level, and it has this probability of sending out the ligand associated with that. And so that's one probability function. And the other one would be like, okay, so this cell receives that ligand at this probability, and you kind of align those and use that to find the most effective way to transport one distribution into another. So that's one thing that we use as input. And then the other thing is space flow, which is model that is able to create a consistent low dimensional embedding of segmentation. So pretty much it takes a tissue and kind of maps out the histology in a way using segmentation. And looking at gene expression and spatial coordinates, it's able to kind of segment like cluster your tissue in a way that these structures are supposed to be like identified through that. So my method pretty much takes the comet inferred interactions along with the space flow domain segmentation, and it kind of takes a four-pronged approach into how we can quantify this. And it's like step by step. So first would be the hierarchical segmentation. So pretty much from the space flow domains, I'll explain an example in a second, but through the space flow domains, you'll have these different regions of the tissue identified as potentially like being a structure or stroma or something like that. And we can further break them down into smaller units I call modules based on signaling. So maybe a lobular unit in one region of the tissue is different from a lobular unit in another region of the tissue. And so those two may communicate differently with its surroundings, so they would be two distinct groups rather than one based on gene expression. So they might be transcriptionally similar, or they might be similar structures, but they would segment differently through the hierarchy. So that's the first thing. And then after that, using those modules, we would calculate a term we deem as flux, which would quantify the information flow from a module to its surroundings and its surroundings to the module and kind of vice versa. So it kind of quantifies the information flow between structure and stroma. Um, then after that, we wanted to look at a multi-step relay. So for example, one cell type signals another, which then as a result would signal another. And so looking at those types of cascades, can we find them in the tissue? So for example, if you want to find a specific relay, where in the tissue is that located? How much stronger is one relay in one part of the tissue with another part of the tissue, and so on and so forth. And then kind of the all tie-in thing for all of this. So these kind of quantify different things that are happening in the tissue. From that, the main question is the end goal is how do we look at diseased tissue versus healthy tissue, and how can we say one is different from the other? And so I developed a way to kind of put them into latent space using all of the metrics I described earlier to kind of distinguish two tissues. Maybe some regions are similar, some regions are different, and we can look at that. I'll explain more examples of each of these. Again, as a fair warning, these are very preliminary results, so we're still doing biological validation with this stuff. So, yeah, yeah, yeah. Interrupt me, whatever. Yeah, yeah, of course.
So for running your method, we have to run Comet and space flow first, or
Yeah.
Okay.
I mean, I'm planning to because Comet and Spaceflow have a lot of parameters that are like a user would need to define for what they would do. So yeah, so pretty much like my idea is either we can embed it within the tool, but again, I feel like personally, as a user, would probably want to individualize how they're doing each tissue segmentation and how they're figuring out what cells would communicate with another.
Signal flow information with gene expression, the yes, coordinates.
It's based on that, but it's looking more at the cell inference from Comet. Yeah, so pretty much anything inference related is based on Comet, and then actually looking at the localized regions in the tissues from spaceflow. Yeah. Okay. I guess I'll keep going. So I'll just kind of break each step one by one and kind of show an examples. So the first step would be taking these spaceflow domains and breaking them down into smaller regions. And it'll make a lot more sense with an example, but pretty much the steps is we start at the ligand receptor. So each ligand receptor has its own network. So what cell talks to what cell? We create a graph out of that. And pretty much we start at the individual ligand receptor level and look at each domain and say, okay, so how does each ligand receptor kind of interact with each other for each cell? I mean, for each grouping of cells, and can we like kind of combine them through various three levels of clustering? We start at the ligand receptor level, and then after that, we take each individual pathway that would have their own consensus graph, and we would combine them into some sort of bigger graph. And from that, we'd build a hierarchy. So for example, here, for this specific domain, there were 5 modules identified. I'll show you what this actually looks like in the tissue and in when looking at TGF beta, but when looking at Wnt, it actually breaks this region down into 2 separate regions. So like this, we can do this for every single pathway in the tissue of signaling and kind of break it down further. So this example kind of shows a lot better. So this is the tissue we're working with, the breast tissue. So this is from SpaceFlow, the output after using it. We identify 5 domains. And so this area is the skin, so we're not going to be worrying about this. This is epidermal tissue. Um, these gray and blue regions are lobular units, and there's no ducts in this tissue. So when we run the consensus, the hierarchical algorithm that I came up with, we can see domain 0 is completely the stroma. So all of these blues, like dark blue cells, and some various random cells here. Domain 1 kind of focuses more on the epithelial, I'm not epithelial, sorry, epidermal cells. So we're not really interested in those. But domains 2, 3, 4, and 5 are actually each of these units, and they're each colored differently as they're differently localized in the tissue. They're each different lobular units, even though they're all described in SpaceFlow as the same structure. So they differ in communication in some way. So that would give us a very good indication. Also, another thing is that the difference between 2, 3, and 4 and 5 are like different layers of each of the units. So different groupings of cells were grouped differently in SpaceFlow. And so these are different layers kind of the epithelial tissue. Yeah. So from here, ignore the drawing. I'm trying to make a computer one, but from that, we would compute what is the information flow from each of these modules. So like, for example, if these were stacked layers, how does this layer interact with this layer? How does the pink interact with the orange? Kind of like that. Um, and so what I did is I defined a function of flux in which we take each of these modules and we kind of draw an outline with this thing called alpha shape. And pretty much we use the vectors to distinguish whether signal flows in or flows out of different regions of the tissue of the module. And so kind of it just quantifies how does information flow in and out of each of these modules in essence. So a biological example is FGF. So in breast tissue, FGF is a growth factor, and there is very strong involvement in which the stromal fibroblasts send it into the lobular units for the lobules to grow. And this constantly happens in healthy breast tissue, so we use this as like a way of validating whether our information flow was good. And so pretty much from this, we should see that there's significant influx of information from the stroma into the lobular units. Um, so in here, I isolate this region, and you can see there's still some outflux, but the outflux is primarily fibroblasts within the module communicating with fibroblasts outside of the module. So it's still fibroblast communication, but we do see fibroblasts from the outside sending FGF to the basal cells of the epithelium. And we can see a lot stronger influx by the arrows being bigger. That's kind of how I quantify how strong the flow is inward versus outward. And so this is based on net communication. So pretty much the regions that don't show any arrows primarily have more of a balance of flow of inflow versus outflow. And the reason there is even outflow is because the epithelial regions of these also contain fibroblasts, which also might express growth factor that send it back and forth, kind of. So that's the second part. Third part is dealing with cascades. So one cell type communicating with another that communicates with another downstream. And the idea is, can we locate these functional units to see communication in sequence? So an example of this is the growth factor relay, which I'll get into in a second. So the overall method is kind of analogous to a Markov model, in which this is like a dummy example. So let's say fibroblasts in the stroma um, have, based on Comet, have a zero point 0.2 inference to the like zero point 0.2 signal strength to send TGF beta to the luminal cells, for example. So that would, if you can convert that into a probability, we can build a sort of network as a Markov model and use them as probabilities. What is the probability of one communicating with another, and then aggregate that over pathways that. So this probability has to happen for this probability to happen. So it kind of becomes a union, and we keep adding to this pathway. So generally, we can figure out the flow of signals. So in this base case example, the maximum probability you can achieve by multiplication would be from fibroblast to basal to vascular endothelial. So the method would output the fibroblast to basal to the vascular endothelial and the ligand receptors involved with that. So that would be like an example of how that works. So in the breast, there is this thing called the basal luminal relay, in which fibroblasts secrete Wnt that activate basal cells, and then the Wnt causes the basal cells to express either AXL, PROS1, or Gas6, and these basal cells use that to activate luminal progenitor cells. And so this would be the cascade we'd want to find. So the end goal would be fibroblast to basal to luminal, pretty much, and using these pathways. And in this example, I used this region, the same region we looked at earlier. And when you zoom in on these cells and draw out the communication, we can see that there's this region and this region in which, let me zoom in a little bit. Oh, sorry. Um, this region and this region in which fibroblast from the stroma is signaling a basal cell on the epithelium, which is then, so that signal is Wnt, and then the signal from the basal cell to the luminal cell is the gas pathway. So this would be an example of one place that the cascade happens. So more from this, as I keep developing it, is kind of determining whether this is coincidental or causal, because like just because this signals this and this signals this and it's a static tissue doesn't necessarily mean that one cascades to the other. But it's under the inference that if the basal cell expresses the receptor to grab the ligand that's sent from the fibroblast strong enough, then it should be intermediate to the last process. And this is also kind of verified because I looked to see if the fibroblast directly communicates with the luminal cells with one of these pathways, and it doesn't. So this we're slowly building out the argument that this cascade would happen in the tissue. So yeah. So that would be an example of the third component of the method. And then the fourth component would be taking multiple samples and trying to compare them. So far, we've only looked at breast tissue and healthy breast tissue, as a matter of fact. So a result of taking the comparisons is we want to compare these lobule units. They should be acting similar, and they should be similar in some way because they're both healthy individuals. So the theory is that most of the lobular units should group together in a latent space. So from each of these like tissues, we obtain similar modules based on cell type composition. So let's say um, different modules have like X percentage fibroblasts, X percentage basal cells. Another module will have similar normalized percentages. And we can use those as a metric of comparison. They have similar cell types. How did their communication different? Like so on and so forth. And then each module from the flux vector fields, is it a source? Is it a sink? Or is it more balanced? Does it receive more signal? Does it send out more for each pathway? So I built a latent space for this in which we combine the ideas of flux, cell type composition, and cell-cell communication in the latent space. So the inputs of this model would be the first input would be the composition percentage. So what cell types are involved? and what percentage of each of these. And then after that, the next input would be intramodular flux. So kind of how does the module itself interact with the surroundings? So the input would be modules rather than individual cells, if that clears things up a little bit. And then the third component of the model would be the intermodular communication. So inside the module, how is signal sent? Is there more signals received, some sent, some received within the module? So how much activity is happening in these modules? And you can and then so the goal of this would be each of these with it would be an individual feature, and we'll multiply that by the number of pathways we have. So we should have a pretty robust model. And this approach allows you not just to compare two tissues, but let's say you run a bunch of samples through spatial data, and you're looking at maybe 10 healthy cases and 10 diseased cases of the same region. You can put them all into this latent space and kind of observe the similarities. So for this example, I used two tissues. So the tissues were obtained by different regions of the breast. So it's the same person that was that got the that we got the samples from, and they're from different regions. And so both of these regions have like the same. They have lobules and they have stroma. So those are the two things we're trying to compare in this case. And so in this plot, this is the latent space as a result of the model. We're still trying to improve it in terms of its accuracy and the architecture to make it more efficient. But again, you can see that there's very few samples and like very few modules that were involved. So I think adding more tissues should be able to help with the accuracy of this. And the red lines um, connecting different dots on this plot indicate high similarity in terms of their cell type composition. And the moderately similar ones are with the gray dashed lines. And so when we look at the actual tissue, these are two different, so these are the two tissues I was comparing. They both have these lobular units classified by these things. But when I was looking at the overall results of the model, I noticed that most of the lobular units were very close in proximity to each other in the latent space, which is a good sign. That means that um, the model assumes that they are very similar. But there was one pair that was labeled as divergent, which was these two. Um, these two, which this one correlates with this, and this one correlates with this. And I drew them out on the tissue where they're located. And the reason that they seem to kind of be more far apart on the latent space is because of flux. So for this example, I just use FGF as the pathway example. And the blue module over here, which is indicated by this, has a slight outflux, actually closer to no flux, where the net communication is very balanced, versus in this tissue, there's significant influx, which would indicate that this region may be in the process of growing a little more compared to this region, which would be more kind of mature. And so I also do a feature importance test to see which features kind of separate regions, like different modules in the latent space. And the top two features were the flux and the B cell percentage composition. So that's why they're indicated by a gray arrow versus a red arrow. I mean, a gray line versus a red line, indicating that the B cell percent composition was that significant that caused it to separate that much in the latent space. So yeah, so those are the kind of components of the method that we were trying to develop. Future directions include improving the latent space validation error and separation. There's a big jumble kind of here, and there's not as clear of a separation. And kind of figuring out, comparing it to PCA and seeing like how can we get these things to separate and then add more tissue, add more samples to it should be able to increase reduce the error that we're having. And then performing more biological validation, looking at common pathways that we should be finding, common flux patterns that we should be looking at, and then more common relays that might be happening. And then we could test this on different types of spatial data, so like StereoSeq, Merfish, Visium, stuff like that. And then the end all goal is eventually we have diseased patients versus healthy patients already readily available for Xenium, specifically in breast cancer. If you have the BRCA1 mutation, you're more prone to get breast cancer. And then so we have 6 samples of healthy versus people who develop breast cancer as a result of the BRCA mutation. And so that would be our new test set that we would use once we can validate this in healthy. So yeah, as acknowledgements, big thanks to Dr. Kai Kessenbrock, Qing Ni, and Axel for helping me through this project and kind of serving as my mentors through this. So yeah, thank you.
I had a question on the last 2 slides.
Yeah.
So yeah, this one and the last one. So I saw that you had a way, great talk by the way, really cool stuff. So I saw that you had a metric for how similar your two tissues, I guess, were to each other. And then one of your conclusions was that the B cell percentage was kind of like what's causing that difference a little bit. What was the actual proportions of the cells in those two tissues?
Oh, I forgot to write that down. Okay, so this one is 80 and this is 110.
For the B cell composition?
Like overall cells. But the B cell specific, I have to look into that.
So like, yeah, like B cell versus any other cell type in that same tissue section.
Yeah, I have to double check that, but yeah, that was one of the main features. So it's more like we have to see if that would make sense and then see like maybe it's a misclassified label, something like that.
That's a bunch of questions.
All right.
So this is pretty cool that you're trying to see if based on FGF, these lobular units kind of are in the same latent space, but don't you think that maybe just because you're only considering FGF, maybe that's why sometimes you have divergence, like you need more flux of pathways to say, okay, yeah, these two are definitely like closer together.
Yeah, I mentioned this earlier in the model architecture. The goal is eventually to add more and more pathways. So like eventually we would add all the pathways that could be represented to make this more holistic. But right now my focus is trying to work out the architecture to see if I can improve the error. And so far, adding more pathways, I've gone from 1 to 4, have not really improved it that much. So I'm looking more into if I add another sample, would that improve it? And then if I do a combination of the two, would that improve it further?
So how you talked about doing scans. Are you just like using those as an example, or there's like a reason?
No, there's a biological reason. That's why I use those. I described this earlier, but a pretty common relay in the epithelial tissue of the lobular units is the basal luminal relay. So it activates these luminal progenitors using this signaling relay that is well documented in mammary gland development and growth. So pretty much helps grow the like kind of lobular units in this.
I asked you because, well, first, I want to know if there's any similarity to like cell chat and neuron chat. It's like them heavily, but in my samples, gas like showed up like severely being different from male to female. And I saw it here, I was like, wow. Then I'm working on Alzheimer's.
Oh, okay.
So like there was a difference between like control samples and AD samples, and then mentioned, I was like, oh, okay, there's something about the biological difference that showed up in my samples and yours. But yeah, I was just also curious if there's any inspiration from cell chat or neuron chat for your method.
I think like kind of cell chat in a way because it's more for like single cell looking at how things interact, and then now I'm just kind of expanding that to spatial.
So it's using like the database that it's the like.
Yeah, it's a cell chat database, I'm pretty sure.
Okay. And then last question. What size of data set, like how many cells per genes do you think like your method can handle?
Yeah, that's a good question. So right now I've genes wise, so xenium, these have been xenium samples, but I mentioned earlier how you have a very limited gene panel. So we actually have matching single cell for the same tissue. So we sequenced it twice. We sequenced it for single cell and then we sequenced it for spatial. Um, and so we had matching sets. So you're able to impute using this package called Spage that kind of looks for matching anchors and kind of like you can figure out what cells are most similar in the spatial data compared to the single cell data and kind of use that to infer what are the gene numbers for that. So I believe in this set, I'm pretty sure I filtered it down a little bit. It's about like, or I'm not sure. It was like 1300, no, 13,000 of, no, sorry, not 13,000. It was like 8,000 genes and I can't, each tissue is different on how many cells, so I can't remember the exact counts. But yeah.
Or as you work more on tools.
Yeah, so far, efficiency wise, the only thing that takes like a significant amount of time is the flux calculation because it's looking at individual cells within a module and then stromal cells surrounding it. So it's a lot more heavy computationally, as well as figuring out how you can draw boundaries around these regions. So that would be the only part. Everything else is pretty efficient. Yeah.
You know, to like have this as majority of work of your dissertation or?
I think so, yeah.
Soon for us.
Probably not soon, but
Like many months.
yeah. I think it's been coming along pretty fast, but now we're really honing in on the biological validation and kind of like working around some of the kinks. Because right now I'm looking into other tissues and I'm noticing some issues, and it's allowing me to make corrections and make improvements to my algorithm. And so, yeah, once we get all that kind of worked out, then we'd start kind of putting it together.
Speaking of biology, I have a question for you.
Yeah.
Is this adult tissue?
Actually, I'm pretty sure it is. Yes.
And if it is, do you see memory gland growth in the adult in the stasis?
I think premenopause.
So are these premenopause?
I think
tissue?
so. I have to double check, but I'm pretty sure. This was given to me a very long time back, so. All right. There's no other questions.
There's a question in the chat.
Oh, shoot.
Well, there's at least a notification in the chat.
Great talk. Oh. Thank you. All right. Cool. Thank you.